Welcome to this Twist-hosted webinar, Writing the Future of Cell Therapies using the Twist Biopharma Library of Libraries. My name is Chris Thorne. I am Senior Manager of Field Marketing here at Twist Bioscience. Before we begin, just a few points of housekeeping. You'll notice that you're muted, but if you have questions, please do input them into the Q&A box, and we'll be aiming to tackle them after the presentation. Also, following the presentation, we'll be sending you a short survey, and we'd be immensely grateful for your feedback as it helps us improve. Okay, today's webinar. Our speaker today is our very own Aaron Sato, Twist Bioscience CSO. Aaron is a proven biologics leader, adept at managing teams to discover novel, first-in-class antibody therapeutics. Prior to Twist, Aaron served as CSO of Lake Pharma, leading the California Antibody Center, which discovers novel antibody therapeutics for its clients. He also oversaw all discovery research functions, both as VP of Protein Science at Surrozen and previously as VP of Research at Sutro Biopharma. With that, welcome, Aaron.
Hi, my name is Aaron Sato, and I'm the CSO of Twist Bioscience. And today I'm going to talk to you about how we're writing the future of cell therapies using the Twist Biopharma Library of Libraries. I'm always asked, why Twist Biopharma? What kind of sets you apart from your competitors? And it's really a three-pronged approach. The first is that we take a very synthetic approach to how we do library generation and antibody discovery. And that really allows us to be target agnostic, really helps us decrease the overall risk, and allows us to develop therapeutics and diagnostic antibodies against almost any target. Second, all of our antibodies are fully human, and we're extremely quick in how we discover and bring those antibodies to the market. And that allows us to de-risk our antibody libraries through making sure that all the antibodies that come out of the libraries are highly developable, and really helps our partners avoid costly and time-consuming rework, and enables them the greatest probability of success. And then finally, we have various ways to work with us, all kinds of different partnering options, from licensing out those libraries to folks to use, or actually doing the work in-house within Twist using our expert team of antibody engineers, as well as even licensing out some leads that we already have in-house. So we know that the race is on to find the best novel biologic drug candidates, and really we want to help others find an advantage to help them discover, optimize, and develop drugs very quickly using the tools as well as services within Twist Biopharma. So we really see Twist Biopharma as basically an elevated division of the company where we can basically use all these amazing high-impact DNA tools to enable antibody discovery and antibody optimization. And as you'll see in a second, we'll also take advantage of many of the library tools to help others, not only in the biologics antibody space, but also in the cell therapy space to help them discover and optimize their particular cell therapies. And so both biopharma as well as data storage are divisions of Twist Bioscience, where we again use those amazing products of Twist for specific applications. I always say the best companies out there really understand the one thing they're really good at. And for Twist, that's our amazing ability to print DNA. So we have this amazing silicon chip where we can actually print up to 1 million individual oligos up to 300 base pairs in length. And we use that as a system to create oligo pools anywhere from, say, tens and hundreds all the way up to 1 million different oligos in a pool. And as you'll see in a second, we can use those oligo pools to generate all kinds of custom DNA products. And as I said before, Twist Biopharma, a division of Twist Bioscience, we use these amazing products derived from the silicon array to help others discover and optimize antibodies and really increase their chance of clinical success. As I said before, it's really three main advantages of Twist Biopharma. It's really the breadth of what we can offer in terms of our library of libraries against a whole host of validated targets. And again, our competitors are oftentimes restricted to using maybe one library, maybe a couple libraries, and that's really because making a library, as you may know, is just really hard. But because, again, our expertise resides in DNA synthesis as well as DNA manipulation to create libraries, we can create high quality, highly validated libraries very quickly. Second is really the quality of those libraries. Even though we make synthetic libraries, since we can make explicit sequences in oligo pools, we can actually make a fully human synthetic library that contains natural human sequence. So even though it's synthetic in origin, all the antibodies that come out look just like any antibody that would be derived from a human. And then finally, Twist is an amazing place for automation. We've automated the entire process of DNA synthesis. And so as a result, we have a lot of expertise in automation, and we actually have applied a lot of those learnings in automation to the antibody discovery and optimization process. So again, we really see Twist Biopharma is helping you to really drug the undruggable. We have fully human antibody libraries. We're very focused on specific sequence spaces. In particular, we have a whole host of libraries focused on difficult drug targets like GPCRs and ion channels and carbohydrates. And as you'll see in a second, we have a broad array of different libraries. There's a lot of advantages to using oligo pools derived from the silicon array to generate libraries. And in particular, in looking at antibody libraries, shown on the right are all the different advantages that you can see by using oligo pools for this application. The one I love to talk about is CDR length variation. Since we're making different lengths of oligos in a pool, we can very easily vary the length of any individual CDR in that library. This is actually really difficult to do with other degenerate oligos or trim-based oligos, and so it's a really fantastic advantage of using oligo pools. The other one I really love to cite is the base-by-base precision that we can apply to what amino acids are allowed at each position in a CDR loop. Again, we can explicitly synthesize sequences in those CDR loops, and we can control the distribution of amino acids as well as the codon usage for those amino acids as we build the library. So as I mentioned before, Twist Biopharma, we're a division of Twist Bioscience, really focused on helping others in the early stages of biologics drug discovery, from antibody discovery through optimization, to help them in their biologics drug discovery workflow, as well as you'll see in a second, to help them discover novel CAR Ts, as well as novel TCR-based therapies. On the antibody discovery side, we're very focused on helping others by creating a whole wide array of different antibody phage display libraries. We call it our library of libraries, and they range from different formats from single-chain FVs to VHH single domains to even fully human Fab fragment antibody phage libraries. And then on the right, we also can optimize antibodies. We can take any antibody derived from any source, whether it's murine all the way to fully human. We actually can pipe it into our custom software that has a very large human NGS database of antibody sequence as part of it. And then we can use those sequences to design a very focused optimization library around your specific lead. If your antibody is actually mouse-derived, we also can humanize the antibody as we do that process to give you a fully human antibody as the output from that optimization workflow. And again, our process is very streamlined, as I talked about before, and very fast. We can, again, select your specific target using a whole host of different antibody phage libraries I showed before. We oftentimes will pool libraries based on the different formats, from Fab to single chain to VHH. We can do cell or bead-based panning using recombinant protein. If it's a protein-based target, we can do clonal ELISAs to pull out all of our specific hits and then sequence those using Sanger sequencing. If it's a cell-based target, oftentimes we'll do NGS analysis at every round and look for enriched clones. It doesn't mean we can't do that also for the protein-based panning, and that both of those sources of sequences allows us to come up with a whole host of sequences to reformat to full IgG. We can make and purify them through our high throughput IgG offering, and then we can put those antibodies through a whole host of different binding and functional assays. So as I mentioned before, our libraries really differentiates us and sets us apart from a lot of our competitors. We have 18 different libraries, and that number continues to grow month after month at Twist. And so we really pride ourselves on creating high-quality antibody phage display libraries that are really focused on different sequence spaces to allow us to find antibodies to a multitude of epitopes on different targets. In terms of how we build a library, it's again takes advantage of the power of oligo pools. If you think about an antibody domain, say a heavy chain, it's basically a series of 3 loops or CDR loops. We can make an oligo pool that encodes for human CDR diversity in each of those different loops, and then we can PCR them together in the context of a fully human germline framework. Since we're using explicit sequences derived from human antibody databases, our library are fully human. I also love to cite that we can often take out sequences as we build these libraries as well. For example, we can take out any CDR polymerization to cleavage, deamidation, or glycosylation sites. And then finally, if we choose to, I won't focus on it today, we can actually include motifs in those CDR sequences as well. And that's actually key and central to many of our difficult to drug libraries focused on GPCRs and also on ion channels. So now I'll go through a brief review of one of our types of libraries called our VHH single domain libraries. The reason why I'll focus on these today is they're actually really useful for our CAR-T based collaborations, as you'll see in a second. So we actually have 4 different VHH single domain libraries here at Twist. They actually started from a single design set of 3,000 different llama sequences that all had explicit antigenic specificity and that they all bound one specific antigen. We use those 3,000 VHHs to design the 4 libraries I'll talk about now. The first one is we call our VHH ratio library. It's built off of a consensus llama framework based on those 3,000 VHHs. And then we actually created oligo pools that encode for diversity that mimics the diversity seen in those 3,000 VHHs. So much like a degenerate oligo, but in a much more controlled way, we allowed for these amino acids shown here in CDRs 1, 2, and 3. And we also allowed for length variation, as I cited before, which again was a really huge advantage of using oligo pools. We were able to easily do that in the context of our VHH ratio library. The second library I want to talk about is our VHH shuffle library. Again, it's built off of a consensus llama framework, but here, rather than use mixtures of ratios of different amino acids at each position and trying to mimic that diversity, we just actually use the explicit CDRs derived from those 3,000 VHHs and just shuffle them in different contexts. So much like you would do heavy chain, light chain shuffling to come up with human antibodies that had defined antigenic specificity. Here, we're doing it on a CDR level and coming up with VHHs that bind any target we go after. Another cool thing about this library is because the final diversity of the library is actually greater than the theoretical diversity of the library. We can actually cover the entire theoretical diversity with this specific library. The third library is what we call our VHH H shuffle library. It's just like the library I just showed, the VHH shuffle library, except now we're actually putting it on a humanized GP47 framework. And so again, the idea here is that the VHHs that come out of this library, which again we're shuffling llama CDRs 1, 2s, and 3s on a humanized framework, could potentially be used directly as a human therapeutic. And then finally, our last library, which again is trying to move towards greater and greater human content, we actually replaced the llama CDR3s and the H shuffle library with human heavy chain CDR3s, and we actually inserted an oligo pool of over 2.5 million heavy chain CDR3s derived from another library that we had created previously. This just shows you a little bit of data of the utility of these VHH libraries, where we selected in the libraries against TIGIT, which is an immuno-oncology target. We actually did 5 rounds of panning against TIGIT with the first 3 libraries. And from rounds 3, 4, and 5, we actually came up with about 50 uniques from each different library. Shown here on the right is the monovalent KDs for each of those 50 hits, and broken down into different affinity thresholds. And so you can see that we were able to get affinities in the range of double-digit to triple-digit nanomolar monovalent affinities for TIGIT. And then shown here at the bottom is just the CDR3 length variation between all those hits. And you can see that we're getting very different CDR3 lengths from the 3 different libraries, again indicating we're getting different sequences, potentially different epitopes for binding to TIGIT. So now I'll get into how you can use a lot of these tools within Twist Biopharma, as well as some new offerings that I'll talk about in a second to help out with both adoptive T-cell therapy as well as CAR-T therapy. We know that synthetic synbio tools are actually really important for this space, and we're trying to innovate and create even more tools to help our partners both in adoptive T-cell therapy as well as CAR-T therapy to get to their final construct so they can actually have an impact on disease in patients. The first one is, again, we know that, TCRs are responsible for the activation of T cells in adoptive T cell therapy. And oftentimes, a lot of companies are actively working to actually find the specific alpha beta TCRs that are involved in this activation against oncogenic targets, and specifically both solid as well as liquid tumors. And so what we've been trying to do lately with many of our cell therapy partners is give them DNA tools where they can create libraries of TCR alphas and betas to help them find those specific TCRs that are actually eliciting this response. And so one early access product that we've created recently is actually what we call a combinatorial assembly library, where we can actually make both alphas and betas and link them together with an internal cleavage site. And we make those alphas and betas using non-clonal gene fragments. And we can actually shuffle those alphas and betas together in the context of a single highly diverse library. And we can actually have up to 10,000 different combinations of alphas and betas in this library. And so by doing this, we can create a whole library of different parts, in this case alphas and betas or other TCR gamma delta, for example, combinations. As I said, we can make up to 10 to the fourth different sequence combinations in these libraries. So say, for example, 100 by 100 combinatorial assembly library. And that allows us to explore the sequence space of alphas and betas or gammas and deltas coming out of a patient-derived sample. And then from that, if our partner has a robust screening platform, they can pull out the specific alphas and betas or gammas and deltas that actually elicit that specific T-cell activity. And then on the CAR T side, that actually can take advantage of many of the antibody discovery tools that I talked about before. So again, a CAR T has, of course, a ligand binding domain that goes after a specific target. It has a hinge domain that basically separates that ligand binding domain from the cell surface of cells where we've, again, transfected into T cells. It has a transmembrane domain, and then also, again, a multitude of signaling domains. So again, using this combinatorial assembly-based approach, we can basically make a library of gene fragments encoding for each of those different regions in a CAR T, and then again shuffle them in different contexts to find the optimal pairing of ligand binding domain, hinge domain, transmembrane domain, and signaling domains. And again, that's exactly what we can do now. Of course, on the ligand binding domain side, we can make custom libraries to help you create your own antibody library if you want to come up with your own targeting domains. But I also just showed you that the Twist Biopharma group also has a multitude of scFv and VHH libraries where we can again help you find those targeting domains to then build into your own CAR T. And then, as I said, just said before, we also can then use those targeting domains combined with a multitude of different subunits from, again, binding domain, hinge domain, transmembrane, as well as signaling domains to create this combinatorial assembly library. We can basically shuffle all those different domains and find the optimal pairing of all those different components to give you the optimal activation of T cells. And again, this is an early access product where we can basically create a library up to 10,000 different combinations to help you discover your optimal CAR T. And here's some really exciting data that we actually did in collaboration with another Bay Area company called Serotyny, where they actually had an amazing platform for designing as well as screening these combinations of CAR T constructs. We created several different combinatorial assembly libraries where we, again, varied all these different domains within a CAR T. And then we provided this to Serotyny, and then Serotyny then went through and actually screened using their system for both for activation, proliferation, and cell killing from this library. And from that, depending on what the target was and the signaling domain, what the targeting domain was, they found a multitude of different combinations of all these different domains that led to optimal activation, proliferation, and killing. And so again, it really shows you that there's not one size fits all for one specific CAR T, that depending on the target as well as the target domain that you're using, you can have different combinations of the other components of a CAR T that are actually very important. So just to reiterate, on the CAR T side, again, we can make a library up to 10 to the 4 sequences, but you can use non-clonal gene fragments to make this combinatorial assembly library. And that allows you, if you have a really robust screening platform, to find your optimal CAR T for your specific target. So just to conclude, Twist Biopharma, we're a division of Twist Bioscience, where we offer rapid and efficient biologics discovery services coupled with all the amazing library technologies I just talked about, in particular the antibody discovery libraries, but also the amazing combinatorial assembly libraries I just talked about. So if you're either in the biologics discovery space or in the adoptive T cell or CAR T space, please follow up with us at this contact information here, and we can definitely help you out with any of your needs in this space. And I thank you for your time and attention. I'd be happy to answer any questions.